So one element of this leadership is that we're trying to make a map of human itch subtypes and where there's similarities between conditions with approvals and non-approvals. Um, so we're better trying to understand that. And recently, in a patient who was erythrodermic, we got a patent on a blood test that helped us point to, with flow cytometry, which cytokines were upregulated. And so it's my belief that that is the future of inflammatory skin diseases and itch in general, is that we'll be able to phenotype them properly. And hopefully, from a regulatory angle, we'll be able to conduct biomarker-based trials to be able to get patients what they need based on their phenotype, knowing that there's tremendous diversity and heterogeneity within a single disease, but also appreciating that across diseases, there really is a ton of similarities as well.
Problem right now is that everything about whether medicines are covered or not is based on line diagnoses that have undergone, phase 3 clinical trials. Those are actually pretty rigorous criteria. So they have to be on label for them to be covered. But in reality, a lot of itch conditions over time, post-market, once a drug is already approved, off-label use of those medications demonstrate often very convincing efficacy for those conditions that are not textbook, there are not ICD-11 codes, there are not clear line diagnoses that are well established and that have not been phase 3'd out. So if we can create a system in which we could actually demonstrate sufficient evidence of efficacy without doing a major phase 3 trial necessarily to support coverage of those medications, that would be a huge step forward. Because otherwise, you're just waiting for a pharmaceutical company to develop keen interest in that area and say, we want to go in this area, we're going to invest millions and millions of dollars for another labeled indication. That's made it very clunky in terms of what we can do.
This field is growing. I think we understand better on the molecular genetic levels, what are key drivers for itch. There aren't a lot of biomarkers specifically. Our group has found just in the last and published last year a neuropeptide that is a very highly associated actually with congestive heart failure, brain natriuretic peptide, which we have known for many years in mice models is a important neuropeptide in the periphery. And we saw that this is a very highly correlated to chronic itch of undetermined origin, also an itch of elderly, itch of patients with prurigo nodularis, and the list goes on and on. And we believe that this is a good marker for chronic itch when you don't have a real understanding of the cause. And we've seen that when treatments that reduce itch had an effect on reducing this BNP level. So we believe that that's one of the markers of at least chronic pruritus of undetermined origin, itch in some of the systemic disease like chronic kidney disease. And we're still trying to learn about that. In addition, we know that type 2 cytokines overexpression is highly associated with many types of itch. So testing cytokine levels serum-wise could be possibly, or some of the chemokines could be helpful also in better understanding itch of different types. I believe that we'll in the future know better about specific markers, and it's an evolving area, but this is kind of the beginning of an era that we'll know more.
We're understanding a little bit more now about how the environment can modulate itch. One way is that the GI system, what you eat, can actually directly stimulate itch. You can have activation of the vagal nerves. And our group actually recently did some gut microbiome testing and found different fungal species in certain chronic itch patients as well. And so that certainly, I think, can play a role in itch, as well as this cutaneous microbiome, environmental pollutants as well, that can directly stimulate nerve fibers and activation of those nerves. So I certainly agree that itch is a chronic disease, and I think that spending more time assessing environmental exposures as root causes, also diet may lead to some novel insights on how that may trigger certain polarizations in the environment. And our group recently published a study about groups of older adults who developed chronic itch chronically, and many of these patients had an immune stimulus exposure. This could be a um, event like having COVID or the COVID vaccine or a PD-1 inhibitor for cancer or the shingles vaccine. It's having a shingles episode of the vaccine. And so I do think we need to understand more about these conditions longitudinally and what are some of the associated modifiable risk factors and things you can change in your environment, or really just at the forefront of beginning to understand this. So um, I think that there's tremendous opportunity to shed more light in itch biology.
Yeah, it's an area that we don't know a whole lot about. We have noticed that the environmental kind of changes potentially the allergen environment has contributed a bit more to itch. Whether that's due to global warming or not, I don't know. But there's a lot like that, or whether that's just simply environmental kind of industrial pollutants. Um, I don't know that my personal feeling is that in general, this is not controversial, but processed foods are probably not good, right? Essentially, I think a lot of our food is pickled. That means the proteins that we eat are pickled, which makes it much more recognizable to our immune system in many ways. That's why I think actually food allergies have become just epidemic. I think that could be the case with
I
itch.
can tell you pollutants clearly aggravate patients with atopic eczema, and I don't think that it would be surprising that it could affect other types of chronic itch. We know now better that there are some sensors for pollutants that can activate itch, like AHR, aryl hydrocarbon receptors in the skin. We know that like after fires or smog or a situation where there was a lot of pollution, atopic eczema got worse, both in pediatric patients and in health and adults. I believe that in terms of food, it's more complex. There are conditions. I have patients who say, well, I eat X, Y, Z, and I feel more itchy. So there are some conditions where we know that it is clearly associated, whether there are some urticarial conditions. The answer is a bit complex in terms that we are not everything we eat causes itch. That's what I want the message. And I want a word of caution because I have patients who've been on crazy diets that were recommended by doctors, including there are these some of doctors that deal a lot with integrative type of medicine and testing the patients and put them in on really difficult diets to uh, live with. And I question the yield of that because the majority of these patients really don't have allergy to all those foods that they were mentioned.